Okay, so now let's start the class. Since the last class, I have updated few contents and few figures, so we have to go through again the transcription process. So initially, I thought that we could join the two transcription process in prokaryotes and eukaryotes together, but later I think it would be better if we just separate the two process, the transcription process in eukaryotes and prokaryotes. So as you already know that transcription means DNA transcription. Like it means RNA synthesis, like producing RNA from a DNA. So that RNA could be mRNA and other RNA like rRNA, tRNA, or microRNA. So mostly we'll be dealing with mRNA because it only mRNA is translated into proteins. The other RNAs are not translated, but they have important other functions, biological cellular functions. So, so the result of transcription is not always a RNA. Sorry, that like the RNAs are not always the result of RNAs are not always protein. So that's one important thing. So um, this central dogma we have seen, but this figure I have updated this figure, the central dogma. So this figure is more comprehensive, so that you will get the idea. Like here, the flow of information we know that from DNA to DNA, which is replication, and from DNA to RNA, which is normal. But from RNA to DNA, which is reverse transcription, which is also seen in viruses, and when the host cell is infected with virus, then the virus tries to reproduce itself within the host cell by this reverse transcription process. So this is also um, quite familiar, this reverse transcription, and the usual one is the flow of information from RNA to protein. So this one. So the other flow of information, which is from RNA to RNA, which is seen in RNA viruses, but which is not shown here. So this is the mainly central dogma. So so far we have seen that in the last class, the central dogma, and this codon triple U mainly codes for this amino acid phenylalanine. We have also seen this figure, the process, the overview of gene expression in eukaryotes. So this one, mainly the left side, is the transcription process in eukaryotes. And then after the transcription, the RNA is modified, and then the modified RNA can leave the nucleus and enter in the cytoplasm, where it can carry out its function. It can either translate it or control other cellular process. So gene expression, gene transcription or DNA transcription is the same thing. Actually, DNA is composed of DNAs. So we have also seen this figure last week. So the basic structure of a gene composed of a regulatory sequence and then a promoter where the transcription will start, and then the codons. Codons will form the RNA, and those codons will be translated in the translation, and at last the termination sequence. We have seen this in the last class. So this is new, I guess, this figure. Gene expression in and transcription together in prokaryotes. So most of the um, like if we see closely in the prokaryotes and eukaryotes, most of the processes are the same, but a few steps are different. Like prokaryotes in bacteria, the transcription process is quite simple compared to the eukaryote cell. So this is prokaryotes. So what happens in prokaryotes is that, as you can see, this is a gene. This entire sequence of DNA is a gene, and in in prokaryotes, like two or three genes could be together transcribed. But in eukaryotes, this is not usually seen. Like the prokaryotes are polygenic or multigenic. One that means one RNA uh, could code for different proteins in prokaryotes, but this is like in eukaryotes, just one mRNA codes for one protein, but it takes like other processing to create different versions of the proteins in eukaryotes. So what happens here, like this is like we know that gene expression is composed of two steps. One is the transcription, and then the other one is translation. So it is quite simple. Like this gene is transcribed, and the result of transcription, the immediate product of transcription, is called primary transcript. And in prokaryotes, this primary transcript, which is RNA, this primary transcript is readily translated. So, but in case of eukaryotes, this is not easily translated. It undergoes further processing. So the other codons are same, like the start codon AUG. There are stop codons, three stop codons. So this is polypeptide or protein is synthesized, and when the protein is synthesized, the gene is expressed. In the eukaryotes, we'll be seeing some other different segments within these coding regions, like the encoding zones. But in the prokaryotes, this gene or segments of genes are very simple. And also transcription translation process is quickly done, and there's no further modification. Also, the three stages of transcription, like the first stages, is a start, initiation, initiation. RNA polymerase comes at the promoter. There's promoter sequence, which mainly RNA polymerase and transcription factors mainly recognize the promoter region, and upon binding to the promoter regions, then the polymerase comes and starts transcription. And then the next stage is the elongation. That means the RNA, the synthesized RNA is growing, RNA chain. This is elongation of the chain. And then it's a termination, like there's a stop sequence, termination of the transcription process. Like in general, we can divide the whole transcription process in simple terms, that is initiation, elongation, termination. But this initiation phase is quite complicated, and this termination phase is not clearly understood, like which sequence or at what time the transcription is ended. So there are gene expression regulation proteins like operons, if you heard the term operons, those mainly regulate this termination process. The initiation process mainly uh, controlled by the transcription factors. But in prokaryotes, there is remember that there's simple, there's no transcription factors, just one protein that mainly attached to this RNA polymerase, that is sigma factor. Just remember the protein sigma factor, that is important in prokaryotes. But in case of eukaryotes, there are a lot of transcription factors, different transcription factors, which mainly start the transcription process, and which mainly those transcription factors mainly recruit the RNA polymerase in the eukaryotes. I will be saying that. So prokaryote is quite simple, not that difficult. Now the eukaryotes comes gene expression and transcription in eukaryotes. So as you've seen in the earlier figure in the prokaryotes, like the that was the coding regions, right? This like after this uh, regulation of translation, that means here the promoter sequence was here in this sorry in this region, in the green. At the end of the green there's promoter, so and there's some additional sequences, those mainly this mainly regulation of translation, like here the ribosomal binding sequence here, and then the coding sequence. Coding means the amino acids are coded within this region. So so this is quite simple, but in case of eukaryotes there are these coding regions are composed of exons and introns. So exon 1, like this is a general structure for a general gene, like there could be 50 exons or 50 introns. Here just three exons shown. But remember that these entire regions, like this exon 1, like from this exon 1 to exon 3, here, this all of the regions is coding regions. Sorry, not coding, but this is like the transcribed regions. That means that from exon 1 to exon 3, all of this is transcribed. But only exons are coded. That means only exons are expressed in the translation. So this is a gene in a eukaryotes. This entire segment of the DNA is a gene. Then after transcription, like I mentioned that the immediate product of transcription is called the primary transcript, because it could convert into mRNA, or it could undergo in the rRNA pathway, rRNA synthesis pathway, or modification of tRNA. So it could turn into a mRNA. So after the modification, we can say, because this is mRNA, so we are calling it a primary transcript. Then this primary transcript undergoes some modification. So if you can recall from the earlier prokaryotes, there were no modifications. So that was simple. But here slightly modifications are done, which is this cap. This cap is new. So this cap is 5 prime. This cap is attached in the 5 prime direction of the RNA, which is a methyl guanosine cap. Yes, methyl guanosine, 7-methyl guanosine. And this at the end portion, you can see like this is new. This A means adenine, and N means any number, like 100, 200, 300. So poly A. This is called poly-A tail, polyadenine tail. So this tail is attached at the 3' end. So all of these modifications have specific functions. Like this tail mainly gives stabilizes the RNA, prevents from degradation from exonuclease or endonuclease other enzymes, and this gap helps in recognition of the splicing site or recognition from like these lysosomes mainly recognize this gap, 5' gap. Also, this modification allows the RNA to be exported from the nucleus to the cytoplasm. So there are channels in the nucleus, the nuclear pore. So there are proteins, those mainly recognize these gaps of RNA and then attach and transport or travel this RNA and export it to into the cytoplasm. So as you can see, in prokaryotes, in the bacteria, there were no nucleus. So the whole region was the cytoplasm, right? So there is no need for travel or transportation of the molecule. That means the ribosomes can quickly attach to the transcription set, like along with the transcription, the the RNA is readily translated. But here eukaryotes, this cannot happen because ribosomes reside outside of the nucleus. So the RNA has to come out of the nucleus to be translated. So as you can see, these brown segments of the RNA are the introns. So brown segments in the DNA was the intron, and this is transcribed. So this one is intron and this one is intron. The green was the exon. So only exons are expressed. That means in the final the maturation, like uh, in the final mature RNA or mature mRNA, is in the mature mRNA, the introns are removed. Only the exons are available in the final mature mRNA. So this is like post-transcriptional modification. So the splicing mechanism, like spliceosomes, mainly works here. They mainly remove these introns and join the exons together. And then it is converted into mRNA, which is mature mRNA. So, so this is mature mRNA. This mainly then uh, travel outside of the nucleus. So then enters in the cytoplasm. In the cytoplasm, it is translated. So that's how mainly one gene expressed in eukaryotes. So, any questions so far? No questions. So now, so the transcription, sorry, the gene expression can be controlled in many stages. So mainly in the transcription phase, the gene expression can be controlled largely, but there could be other stages. So can anyone tell me, like, at which point we can control gene expression of a gene? Like there are several steps within the transcription and translation process. Anyone? Like we have seen that what happens when an RNA is produced. Like if the nuclear pore is blocked, then a certain mRNA cannot come out of the nucleus. That means the gene is not expressed. This is one mechanism. Or if somehow it comes out of the nucleus, suppose it's an mRNA, then what could happen? Like that mRNA could be degraded, right? You done an assignment on the non-coding RNAs. Like if you can recall the microRNA, the function of microRNA, mainly microRNA inhibits the mRNA, right? So after transcription, like an mRNA can be degraded or inhibited. So that is one mechanism of gene expression, controlling gene expression. The other mechanism could be like the protein, like when it's translated into a protein, that protein can be inhibited, or like the expression of the protein can be controlled. So in some many steps, this gene expression can be controlled, but the mainly like if we control the transcription, then there's no need for other mechanism. It mainly controls the whole process. So we'll be seeing that. Like as you can see, this is the first step. This is the DNA or the gene, and the gene is transcribed. This is the primary transcript, and then it modified into a mRNA or other RNA. So in here, in these steps between this DNA to RNA transcript, this is transcription. So here, if we can inhibit this process in the DNA, like the operons or other complexes, mainly bind to the promoter region of a gene, so that if it is bound, that means the polymerase cannot go through, that means the gene expression is blocked. So that is the transcriptional control. So next, this is RNA processing control. That means primary transcript is modified after the transcription. Eukaryotes is modified. So if we can block, or if certain enzymes can block this modification process, like the tail, the poly tail, like the polyadenylation is inhibited. That means that RNA is not could not travel outside of the, or it will be readily degraded. So that is one mechanism of gene expression control. Is the second step. And the next step, the gene could be controlled in expression. That is, the mRNA is like what happens. The mRNA is produced. That means it can then travel outside of the nucleus. That means it can enter the cytoplasm. And in the cytoplasm, like in this phase, like this, suppose this is a nuclear pore, this channel can be inhibited or blocked. So this is another control mechanism, mRNA transport and localization control. So if somehow it has gotten outside, then it can be further control, like the degradation. This mRNA can be degraded, like this one, this mRNA degradation control. So this mRNA can be degraded by other RNAs, like the microRNAs or siRNAs. So mRNA degradation control, and then suppose the mRNA is not inhibited, like this is translated into protein, then translation control. In here we can block the ribosomes. If the ribosomes is blocked, then this mRNA is not translated into proteins. Somehow it is translated into proteins, then the protein can be further, the function of the protein can further um, blocked or inhibited. Like the protein after translation, it undergoes folding, like conformation of change, 3D structure, folding, then after folding it is functional, and some other modification. So those modification folding, like the the heat shock proteins, chaperones, molecular chaperones mainly done this folding. So those chaperones mainly control this gene expression at this stage, protein degradation control. And also there is like the proteins are degraded by other mechanism, which is the proteasomal ubiquitin pathway. So here the gene gene expression can be controlled or inhibited at this stage. Like the proteins can be degraded. The proteasome mainly fragments all the amino acids and recycles these amino acids. And the other one is the protein activity control. So like the modifications can be done. Like one modifications can turn into a inactive to active proteins, so it can go reverse. Like the modification can control the activity. The modification can render the protein into a inactive one from an active one. So these are the seven different steps where the gene expression can be controlled. But mainly, like if you control at the very first, then this all the unnecessary, like the mRNA modification, translation, all the unnecessary steps can be inhibited if we just can control at the first step, right? So here the it's all written here in simple words. So any questions from here? Which control of the gene expression? Like, are you getting this? Like I'm talking solely, so this is not quite interactive. If you can respond like yes, you are getting, then it would be better on my side. Like, is it difficult or is it easy or is it like okay? Any question
Yes, sir.
from
So
your
far,
side?
I've understood everything.
Okay, that's great. So gene expression is important because um, after protein, like all the research and all the development is happening in the earlier decades. All the focus were on the proteins because proteins are interesting. They can be visualized or they can be seen in different forms. But within this, when the focus has shifted into now RNAs, because we know that we only knew that only the mRNAs can be translated into proteins and proteins carry out all the functions. Like we can virtually, what we see, everything is protein. So, but now we can see that mainly the RNAs mainly control the expression of the protein. So there are interesting RNAs. So these RNAs are quite interesting. So this focus is shifting toward the RNAs, transcriptomics and epigenetics and epigenomics, this side. So now we now start the initiation process of transcription, like how this transcription starts. Like I mentioned, this one protein sigma factor in the bacteria. In bacteria, this sigma factor along with the RNA polymerase, this one is important, this sigma factor. So like this is this is the gene or DNA, and then within the DNA there is a promoter regions for a gene. Promoter regions mainly start the transcription process, and RNA polymerase mainly recognize this promoter or mainly start from here. So the bacterial polymerase contains this sigma factor, this uh, uh, yellow circle protein sigma factor. So what happens next? Then this, like if you can see, the promoter was this one, this green. Here the green is not that much visible, but here you can see this. The sigma factor mainly binds at this promoter, and then the sigma factor is released. So when the sigma factor is released, then the RNA polymerase starts to move and synthesize the RNA. So up to this point, the terminator point, the RNA polymerase will synthesize the RNA strand. So as you can see, the sigma factor is released. So at first, the sigma factor resides within, like these two are joined together, the polymerase and sigma factor. And then when the sigma factor reaches the promoter, then it recognizes the promoter regions, and then the sigma factor is released or dissociate from the RNA polymerase, and then the RNA polymerase then moves and starts synthesizing. And as you can see, um, up to this point, when the polymerase reaches this red sequence terminator, it stops the transcription, and then it dissociates itself from the strand, DNA strand, and again the sigma factor joined together rebinds. The sigma factor rebinds to the polymerase. So it is simple in the bacteria, it is simple, but in case of eukaryotes, there are a lot of other proteins, transcription factors involved in this initiation process. Now we'll be seeing that in eukaryotes. So we know that there's a special sequence in the promoter regions, the TATA repeats. Those TATA repeats are called Tata box. And these Tata box mainly the transcription factors recognizes these TATA repeats sequences and binds to this portion, and like here this transcription factor 2D, TF2D, this mainly comes, and within this TF2D there's this shaded region. You can see this one is the Tata box binding region, you can say. So within this region, it will attach to the Tata box here, the green one, as you can see, the shaded one and this green one. So this is the first step in the transcription of the eukaryotes. So next, whenever this binds, this transcription factor 2D binds to the Tata box, this mainly uh, distort or change the shape of the whole promoter regions, and this by this change it mainly recruits other transcription factors. As you can see, next the transcription factor 2B, this comes and attach with the previous transcription factor, which is the transcription factor 2D. And when this one comes, this transcription factor 2D, then this duplex or this complex mainly recruits the other transcription factor and RNA polymerase. So as you can see, this is the RNA polymerase, and within the RNA polymerase there are other factors, other general transcription factors, and transcription factor 2E, 2H. All this comes and bind to the promoter region. So, and then that means the transcription factor comes first in the eukaryotes. Those transcription factors changes shape and recruits further other transcription factors and RNA polymerase, and then RNA polymerase start to synthesize. Before that, this RNA polymer, like this transcription factor mainly released from this complex, initiation complex. Only the RNA polymerase stays here. The other one is released or dissociates. Just RNA polymerase and this this one, this pink one remains together. This pink is the 2H transcription of the 2H, and this RNA polymerase stays. No, sorry, not pink one. No. This one, the first one is EF2D. This one stays together, and the RNA polymerase stays together, and then this mainly, as you can see, this direction, this goes on up to the terminator point. So this is how mainly the transcription process starts in the eukaryotes. As you can see, the UTP, ATP, like the ribonucleotides mainly joins, and the general transcription factors like this one all are released. So this is the initiation process. This one, the structure of the promoter, we have seen in the last class. The pattern of the promoter, like the regulatory region and the data box, and this transcription start site mainly composed of these two pyrimidines, this adenine, cytosine, and 5 pyrimidines downstream to the adenine, and 2 pyrimidines upstream to the adenine. So this is the common structure mainly found most of the promoter of the genes of the eukaryotes. And these regulatory regions, the hallmarks is this GC and CAT boxes. That means those repeats are found within the 50 and 100 upstream nucleotides from the first transcriptional starts at this first one. So regulatory regions mainly control the whole process, like enhance or silence this transcription. All are written here. So just 1 minute break. We'll join after 1 minute. So in the transcription, the DNA had two strands, right? So and two strands are complementary. So if the one strand, like if the gene is resides in the one strand, that means the other strand is complementary. So if you just imagine that, like the like in the two strands of the DNA, the transcribed RNA is complementary to the other strand of the DNA. So, like, we'll be seeing that how this mainly works. Like the template strand, like when which like the RNA polymerase uses that strand for synthesis, that is the template strand. And the other strand, those mainly remains empty. That means the RNA polymerase don't transcribe. That is the non-template strand. So as you can see, the two strands, like imagine this one is the gene, this portion is transcribed. So like portion that means two strands is not transcribed, just one strand. So the gene could be at the upper strand or the lower strand or on the both strands. So just at one time, one polymerase is like one gene is transcribed at one time. So if you consider like one gene in the upper strand, this is the template strand. That means the gene is this strand. So as the RNA polymerase will synthesize this strand, and the resulting RNA will be complementary to this strand. That means the RNA would be the first nucleotide would be the G, and this G, the second nucleotide would be C. That means if you consider that other DNA strand, this one is also complementary to this one, the gene. So that means like the resultant RNA is equivalent to this one, non-template strand. So this is quite interesting if you just think that like after transcription, if that is your mRNA and template strand, the DNA template strand is this one, like this one is transcribed, and the other non-template strand is this one. This one is complementary. Both are complementary. Yeah. So the sequence of the mRNA would be equivalent to this one, like the non-template strand. As you can see, like the mRNA is complementary. The mRNA will be complementary to the template strand, right? So as if you focus on the non-template strand, so the first nucleotide is G in the non-template strand, and the first nucleotide of the mRNA is G. That means same. It means equivalent. C, A, and in the place of T, you know that in the DNA there's thymine, but in the RNA that is uracil, so that is equivalent. So except the T, all other nucleotides will be same. So this is like the equivalent of the non-template strand. So this is what intended here, the template strand and the non-template strand in the transcription process of the gene. Also the non-template strand is called the coding strand, and the non-template strand also called the sense, and the template strand is called the entry sense. Non-template is called coding strand because ultimately, the resultant mRNA sequence is equivalent to the non-template strand. Always in the scientific writing of mRNA or any sequence of the DNA or RNA, we always write from the 5' end to the 3' end. No matter what is your sequence, if it is given in the reverse direction, you have to write from the 5 to 3' end. So now, like in the earlier, like there, if that is the sequence of a chromosome, there could be multiple genes. But here could be one gene, here could be one gene, or like 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 the upper strand in the upper strand there could be one gene, and in the lower strand within the same region could be another gene. So multiple gene. In case of multiple genes, the transcription mainly works like it could go both directions depending on the upper strand or the lower strand. So if that's the chromosome and gene A, the first gene is this one, like the like the DNA polymerase, the RNA polymerase also travels from this direction, 3 to 5 direction, movement of the RNA polymerase. So so if you can see the 3 to 5 direction is like the transcription is on rightward. If the template strand is this one, if the template strand is the uh, like the RNA polymerase will go from left to rightward, 3 to 5 prime. In case of other gene, like if the gene resides in the lower strand, or if the template strand is the lower strand, then the direction will become like 3 to 5, this direction rightward. But there could be like the gene can be in the upper strand. Like in case of this gene, the gene C, this gene mainly resides in the upper strand. So the transcription direction would be leftward here because RNA polymerase goes 3 to 5 direction. So from here to here. So that means a gene could be on the both strands. So yes, next, like there are questions, like one question is added, and there are two questions in the last class. The new one is the middle one. No, not middle one. Um, which one is the new one? Mutation change. Stop.
The last one.
That's the last one. Yes, the promoter. So the mutation has changed the promoter sequence of a gene. This mutation affects the so can anyone try to explain this one, the last one? If you can understand. So now actually it depends. Like the mutation, like if it alters the nature of the mutation, like if the mutation uh, changes the situation in a way that mainly like the RNA polymerase cannot recognize the promoter or the transcription factors cannot recognize the promoter, then your answer will be correct. But if in case of explain, you have to explain the both case. Like what could happen if a mutation? I haven't mentioned the type of mutation or the mutation has changed the promoter sequence in what direction or in like it rendered the promoter in what way, good way or bad way. So you have to explain the both ways in brief. So usually that's correct. The promoter sequence change in the promoter mainly and RNA polymerase and transcription factors cannot recognize that promoter. So the gene expression of the RNA is not expressed, is not transcribed. So there are in the bacteria there are one polymerase that mainly codes for transcribe all the RNAs, but in the eukaryotes there are mainly three polymerases for RNA polymerase 1, 2, 3. And in plants there are two others, 4 and 5. But in case of humans, animals, mammals, there are one, two, three. So one mainly codes for the transfer RNA or ribosomal RNA. The ribosomal RNA. So one transfers the ribosomal RNA except the 5'7", 5S RNA, the small one. 5S is the least, very short RNA. So RNA polymerase 1 synthesizes all the ribosomal RNAs except the 5S1. And the RNA polymerase 2, which mainly goes for the mRNA that we have seen so far, the mRNA. And it also transcribes the snRNAs, that means the spliceosome, the snRNA, a small nucleotides that mainly forms the spliceosome. And this spliceosome mainly then splices the mRNAs. So, and like the easy way is like RNA polymerase 1 transcribe the rRNAs and the RNA polymerase 3 transcribe the tRNA, transfer RNA. And the two RNA polymerase 2 mainly codes for the all the other RNAs. Like this 5S RNA is transcribed by the RNA polymerase 3, the transfer RNA, and this 5S mainly transcribed by the RNA polymerase 3, tRNA and the 5S RNA. So in a lesser extent, the RNA polymerase 3 also transcribes the snRNAs and a few non-coding RNAs, but that is not uh, usually seen. But it is lesser extent. You can just remember this one: the fibers and transfer RNA and the ribosomal RNA, except the fibers RNA. And rest of the RNA are transferred by the polymers too. And the function of the RNAs, different RNAs here. Like the mRNA, we know that it is mainly translated into proteins. tRNAs helps in the transferring of the amino acids and the translation of the mRNA. rRNA, which mainly forms the ribosomal parts and helps in the translation process. snRNAs, small nuclear RNAs, forms the spliceosomes, which mainly splits the introns out of the mRNA, primary transcript of the mRNA or pre-mRNA. The microRNA is quite important. It is 20 to 20 nucleotides long. It mainly blocks the mRNAs. That means it mainly blocks the protein synthesis of the gene or protein synthesis of the transcript mRNA. So that's mainly it. So yeah, we will end here. The next week we will be seeing the modifications, view, capping, poly tail, and then splicing, alternative splicing, and then RNA editing. The uracil can be converted into xanthin. So those are quite new. Like those are not seen in frequently we don't see that. But this is like as the science is progressing, we're seeing a lot of new things and our idea is changing every day. So yeah, I'm going to end here.